I am Keegan Sawyer, the director of this project on gene drives and non-human organisms. Thank you everyone for joining today's webinar. Jim Collins, who is on the phone as well, is co-chair for this committee, and he will welcome you as well.
Hello everyone. It's a pleasure to have you with us today. I'm Jim Collins, as Keegan said, at Arizona State University and co-chair of this Committee on Gene Drives for the National Academy. We want to thank you for joining today's webinar on the status and directions of basic research on gene drives. It's really interesting and fascinating new research area. Today's talk is going to be given by Dr. Ethan Bier from UC San Diego. It's an information gathering webinar for the committee, and therefore only committee members will be asking questions during the Q&A period. Dr. Bier will also be joined by his research colleague, Dr. Valentino Gantz, to answer committee questions about their presentations. So with that, I'll turn this over to the two of them and we can get started.
Thank you. And I'd like to thank the committee for giving us an opportunity to provide input to this very important process. So we actually came at this gene drive field from the outside, so we're relatively new to the field. The problem that we had faced was a problem that probably only 4 people in the world cared about, Valentino and myself being 2 of them, which was a problem in evolution and development wherein we wanted to know how a gene regulatory network that was operating in Drosophila melanogaster might similarly be operating in a different, distantly related fly species that had a morphological difference from the Drosophila flies that we study in the lab, and we wanted to understand the basis for that. And the problem that Valentino, who was a graduate student then, he subsequently has graduated and is now a postdoctoral fellow in the lab, the problem that he was facing was one of how to use the CRISPR technology, which he had implemented in melanogaster with great success based on the work of many others, how he could use that to test a very simple hypothesis, which would be that if one took out a specific gene in the gene regulatory network, there should be a certain morphological consequence of that. And the problem was that was not such an easy thing to do in this species that didn't have transgenesis, that didn't have existing mutant alleles. And so he thought of a way of generating mutants in a single step so that might enhance this possibility. And so I'm sure by now you're all very aware of how all this works, so I'm just going to go through it very quickly here. But the essence is that what he thought is based, again, on this CRISPR technology, wherein you have Cas9, the enzyme, that is guided by a gRNA, a guide RNA, that cleaves, makes a double-stranded break, and then there are 2 possible ways that that double-stranded break can be repaired. One, through non-homologous end joining, which it sort of puts a bunch of gum, you could say, in the break, messy, makes indels, and that was certainly not something he was hoping would happen for his experiments. And the other was homology-directed repair, wherein the break is repaired by information from the opposing chromosome, chromosome B in this particular case. So what Valentino thought is that he could sort of co-opt that logic and create a plasmid that has the homology arms flanking the gRNA cut site. And the idea was that the vector would encode both the Cas9 and the gRNA, which would then assemble once expressed. It would cut the DNA at the directed site, and then the homology arms on the plasmid would direct homology-directed repair, if that was the operative pathway, based insertion of the gene cassette. And then once that happened, the idea was that it could happen again from now the genomic source. So the Cas9 and gRNA that are now present on the chromosome could be expressed, cut the opposing chromosome B, and then again the same process would take place. But now, of course, the homology arms span the entire chromosome arms on either side of the insertion site. And the idea was that that could potentially lead to efficient homology-directed repair, sort of basically a biallelic insertion of the cassette. And so he referred to this as the mutagenic chain reaction in analogy to the polymerase chain reaction, in that at each generation it should double the frequency of this allele as a PCR machine doubles at each cycle the template concentration. And so the first gene that he thought of trying this on was in fruit flies, of course, which are the model system that we work with and where the tools are all available. Simon Bullock's lab and others had created a variety of optimized genetic tools for doing this and showing proof of principle in flies. And among those, the yellow locust had emerged as a very good candidate. In addition, the yellow locust was one in which the body pigment could be scored over the entire body, so one in principle could see both the somatic effects as well as the germline effects. And finally, it's known that the yellow locust imparts, mutants in the yellow locust impart an extreme fitness disadvantage to the organism, wherein yellow males are all but prohibited from mating with wild-type females if there is a choice between wild-type males and yellow males. So for all those reasons, the yellow locust seems like an ideal target site to initiate these experiments. And so he built this very, very simple first-generation MCR element, as we refer to it. And the idea was then it would insert as indicated in the top diagram and then now in the lower one here, and that once it was inserted into one chromosome, as I mentioned, it would then copy itself to the other via homology-directed repair. And indeed, this worked far better than certainly I expected that it would, and it performed in the way that this scheme sort of indicates that it might. So if one looks at Mendelian inheritance on the left, the yellow locust is an X-linked trait. Typically, a mutant allele is inherited and a wild-type allele are inherited, the standard Mendelian gene, so wherein then, given that yellow is a recessive allele, if one looks in females, which would be the only one getting the X chromosome from the male, then one should see 50% of the chromosomes are being carrying the mutation, and 100% of the fly should have a wild-type phenotype. Whereas with MCR inheritance, you would expect that the same thing would hold initially. That is, you get one red chromosome and one gray chromosome, but then the red chromosome would convert to gray chromosome, and so now you'd have in females both copies. And so now overall, if you look at the entire population, 50% of the animals, that is males, would be wild type, and that would be the same in any case. But 100% of the females, or 50% of the total, would be mutant as a consequence of that gene conversion. And again, as was published back in mid-March, this indeed works where you can see, if you focus on the line where the females are present. I don't know if you can see this pointer. Can you all see the pointer on the screen? Then you can see that the females, which are the ones that are important, the great majority of them were yellow minus. That's the phenotype up here, and only 8 of them were not. And if you do the arithmetic, that's a 97% transmission, but then, of course, by chance alone, Mendelian chance alone, you'd expect 50% transmission. So if you parse that out, then this little equation here gives you the conversion rate, which is still a pretty respectable 96% in this particular experiment. And then the other thing, of course, to note is that the great majority of those that inherited the element were pretty much full body yellow, and there were some mosaics, but the element was also very active throughout the somatic. So those were the results from that particular experiment, and that has led us then to think of a variety of new strategies that could be used with this system, and some of the ways that it could be used not just as a full MCR, but as what we refer to as split drives, or in our case, we call these copycat elements. Copycat elements in the sense that they can be copied in the presence of a source of Cas9 that is provided in trans. So in contrast to the MCR, which has the Cas9 and the gRNA together between the homology arms, these copycat, or often also referred to as gRNA drives, have only the gRNA between the homology arms. The Cas9 is provided someplace else. This provides, this will probably be the template for which almost all laboratory experiments that use what we refer to as sort of active genetics. That is this process wherein in a Cas9 dependent way you can get conversion of one chromosome to another. This is probably the method that will be used almost exclusively in the lab for almost all practical applications that we can foresee. Of course, there are cases where one will, just to understand the mechanism by which MCRs function and propagate, where one will want to use a full MCR as well, and we'll get at the end to the different ways that one should think of those experiments. And that'll be in a bit that we talk about that. But in any case, if you look at a copycat element which lacks the source of Cas9, they could carry one gRNA or two gRNAs. We've done both kinds of constructs. They work quite well. A single cut causes an insertion. A double cut causes a deletion and then an insertion of the element. And then when you're in the presence of Cas9, that copies from one chromosome to another efficiently. And then when one wishes, one can then cross the Cas9 source out and return back to the standard Mendelian genetics. That's several advantages from a scientific point of view, which is the concern of I think that's the point of greatest interest here. From a scientific point of view, it allows the ultimate control of the experiment. It also is a safer way of doing it. That is, one doesn't have to worry about it being such a dramatic drive as is the case for a full MCR. And I'll get to that when we talk very briefly about some models. So then if you kind of just compare again the standard Mendelian inheritance versus these copycats, which we refer to as allelic pumps, or the MCR, you can see certain very obvious trends. So, for example, if you had a mutation that started off in the population and it was crossed then to wild-type progeny from that point on, at each generation it would go down by 50%, 50%, 25%, 12%. Now, the same trend of decreasing happens with the copycat elements, or again, which function as an allelic pump. But as you can see, it doesn't drop as quickly. And the reason is that every time there is a source of Cas9, as indicated by these circles, in those cases, the element, the copycat element, will be copied efficiently over to the other chromosome. So that it's impossible then for the copycat element to segregate away from the source of Cas9. The opposite isn't true. You can segregate the Cas9 away from the gRNA. So, for example, this chromosome just has the gRNA on it, but not the Cas9. But every time that the chromosome has the Cas9, the gRNA will be on there, assuming, of course, this is at 100% efficient process. So what it really comes down to is that if you say, for example, inoculated a wild-type population with a copycat element at, say, 1%, then at each generation, it would add another 1%. So you go from 1%, 2%, 3%, 4%, 5%, et cetera. Whereas with an MCR, of course, initially it expands in an exponential way. So it would go 1, 2, 4, 6, 8. And then, of course, eventually that tapers off to a logistic growth type of math. But that's the big difference between the two. And again, you can just sort of see that graphically here, where this is taking a seeding frequency or proportion of 1%. So let's say you had a wild-type population of mosquitoes, and you produced and you introduced into that 1% of this transgenic MCR, then you would predict that if it was a fitness-neutral element and it was 100% efficient, again, all the ideal conditions, that it could take over in approximately 10 generations from 1% to nearly 100%. But the copycat element, again, would just be linearly increasing, so that by 10 generations it would be just up to 10%. And so it's a much more gradual change. And I would just point out that the standard techniques that people use in the lab all over today now, where you just have a fixed source of Cas9 and a fixed gRNA, not one that's flanked with homology, also actually tends to grow. And in this case, it grows as the square, in a linear fashion, as the square of the initial seeding. So in this case, it would grow as a factor of 1 in 10,000 at each generation. But what we can see is that if we increase this now, the seeding frequency to 10%, the MCR again now goes very quickly in almost 4 generations. But you can see that the copycat element also is a significant drive at this point when one carries it out to about 20 generations. and moreover that the traditional type of mutagenesis gene actually becomes visible now as a linear increase that is actually identical to what one sees with the seeding frequency at 1/10 of that with the copycat element. So these elements, these methods all have some inherent degree of drive to them. The one that's, of course, the greatest concern is the MCR, but one wants to also be cognizant that copycat elements or gRNA drives that only have a capacity, if seeded at high enough frequencies, to be significant drives. Now, I'd like to move on to the use of these then as in two possible scenarios. One scenario is where one drives some kind of element into a population with the idea of reducing the numbers of the vector insects, like say mosquitoes, killing the mosquitoes, in other words, possibly even eradicating them in a given local population, and that's often referred to as population suppression. And the alternative, one that I personally and Valentino, I believe, would agree as well, favor is population modification, where one does not try to do any harm necessarily to the vector species, but one uses it to drive an element that modifies the phenotype of the population. For example, in the case of malaria, it's just make mosquitoes not carry malaria. So I'd like to start off with the traditional views that have been very, very well initiated and then followed through with detailed modeling by people like Austin Burt, with the idea of population suppression. So the basic idea here is you take an NCR type element or a full active gene drive that is targeted specifically to the germline. So it has no somatic effects at all. And it targets then something that if you knock it out would be either lethal or better yet sterile or grandchildless, according to the modeling. And then as you initiate spreading this, inoculate this into a population, initially the animals that it mates with are all wild-type animals. And in those wild-type animals, because it's only going through the germline, somatically they'll be receptive for the mutation. And so it'll be silent until it builds to a significant enough frequency in the population, say 30 to 50%, at which point animals begin to mate with each other that carry this element. And then just by standard Mendelian genetics, they will, of course, begin to manifest homozygous phenotypes, lethality, sterility, and so forth. But by this point, the bad news for the mosquito is that it's so pervasive in the community that it's hard to stop the spread. And what Austin Burt again has and colleagues have shown by modeling is that if you have a drive efficiency of say 95%, then you'll get an equilibrium frequency of the allele in the population, which again goes to 95%. And this, of course, is taken to an extreme, can lead to either extreme reduction in the population or even to localized, complete eradication of the organism, at least within the local arena. And this is particularly true if multiple efficient MCR-type elements were released at the same time. And so I'd like then to look at the alternative, which is, again, what we prefer, which is population modification. And so in this case, what the idea is you take an effector gene that is under the control, say, of a blood meal, and this is work that several labs have done, but we are in closely collaborating with Tony James at UCI, who has been a leader in this area. And you take, for example, single-chain antibodies, which he has identified, two of which that in combination can make a mosquito completely incapable of propagating the Plasmodium pathogen. And then those effectors are activated by a blood meal. And so what James and colleagues published in their most recent thorough analysis of this kind of element was that if it were coupled with a mechanism for gene spread, then malaria resistance could become a self-sustaining disease control tool. And so he was very excited about this kind of idea of getting essentially a motor to drive this through. And so as soon as we recognized that this method that Valentino had concocted in fruit flies was working as well as it was, we called him up, and he was very excited about the sort of collaborative enterprise that we've then taken on together. And the idea is then one can put the Cas9 and gRNA together with this and have an MCR that then drives this into a population. And both our groups and, as I understand, several others are now proceeding along these lines with some success. So I'd like to then compare the population suppression and modification schemes, give some examples of strengths and weaknesses of them. The advantages, of course, of population suppression are that one can, in principle, eliminate a vector for more than one disease. So, for example, mosquitoes can often carry several different types of plasmodium. They can also carry viruses as well as plasmodium. So, in principle, if one takes care of the vector, then one might be more efficiently eradicating disease in a given area. And, you know, if successful, going all the way to eradication within a local region at any rate, in principle, although I think, as Fred Gould said in his webinar, that's probably unlikely to be the case in typical real world, it could solve the problem permanently. The disadvantages, of course, again, if one assumes that probably unlikely scenario where it would actually lead to the extinction of an organism in a given region, is that it would just do just that. And of course, that is, you know, given all the terrible things that human beings have done to the Earth in terms of exterminating species with global warming and other thoughtless measures, that's certainly something to be concerned about. The other thing that comes again from a lot of work that has been done by modeling, Austin Burton in particular, is that vectors of this kind can create burnouts. If one thinks, for example, of Ebola, a really virulent virus, it can burn itself out under certain conditions. And then that will lead to disconnected populations where, in principle, then the element can diffuse back in and lead to difficulty in controlling the ultimate eradication of the vector. It's also vulnerable to the reintroduction of target species. That is, if you completely eradicate it in a given region, then with time it might come back in, and then there isn't any element. There are no surviving mosquitoes that carry the element, and so you'd have to just do it all over again. So it wouldn't be sustainable. Population modification, on the other hand, can eliminate a single targeted disease, at least with a single MCR. One could imagine MCRs that carry cassettes that work for more than one disease too. That's a possibility. And if successful, it could solve it in a stable fashion because it's always present. And if new mosquitoes diffuse into the population or fly into the population, then they'll get it, and so then they also will not be able to propagate the pathogen. In our view, a very salient and important feature of it is that it does the minimal ecological damage that I think one can imagine in terms of using such drives. And it's not subject to burnout, so that, for example, if you have a local population that is not immediately available for this vector to spread into, then with time there will be some transmission from the stable population to the other population, and you won't see this sort of burnout phenomenon. And then, of course, the disadvantages of it, probably the most important, is that it might require several implementations to eliminate all the different vector diseases. So, for example, you know, a single mosquito can carry more than one plasmodial pathogen. So you'd have to make one for each one, or at least have gene cassettes that were present for each one. And if you wanted then to get rid of viral diseases, you'd have to do that separately. So those are our views of the suppression versus modification. We would favor modification over suppression, but certainly we can understand that there will be conditions under which suppression might be justified. I just want to then also touch very briefly on safety considerations for laboratory experiments. Although Drosophila poses no threat to humans or agriculture, we recognize, of course, right from the very inception of these experiments, in discussions with colleagues at UCSD also, right away at the very beginning when we were starting to think of this, that these elements in principle could spread into the wild. And so we chose to use extremely stringent confinement protocols where we had additional levels of physical containment within the laboratory, and then the flies were all killed before analysis, and all of these flies are being kept in an ACL2 insectary that is suitable for example, experiments with malaria or trypanosome infected arthropods. And so we think this was quite in excess to what is actually necessary for containing something like a fruit fly, which is not even considered a vector by the standard measures of vector definition. But nonetheless, we felt that that was important. In addition, we were a contributor, a very active contributor to a group that got together and discussed this idea of how to confine organisms to the laboratory that contain these, and we reached a consensus that was a very prolonged and extensive discussion that I believe lays out all the important methods that are in place today that researchers can use to make sure that they carry out their experiments in a safe way. And in my view, the most important element of that is that people are simply aware that this is something that needs to be done and needs to be taken very seriously. So again, just to summarize briefly what some of those methods are. If we can consider a full MCR to an endogenous fly gene, that it can then, in principle, copy to the sister chromosome. That could happen in the wild as well. It could spread exponentially or logistically, but at least initially exponentially. And the confinement there that we believe is appropriate is an ACL2 level infector. A split Cas9 or a copycat element, as I mentioned, if seeded at a high enough frequency, can be a drive. But it spreads linearly, and of course, if there's any selection against it, the selection will act in an exponential or logistic fashion against it, and so it will die out very quickly if there is any selection against it at all. Nonetheless, one should be careful, and I would suggest perhaps that standard laboratory methods with some dedicated room perhaps, or at least a very clearly delineated region of the room where it's carried out, and perhaps additional confinement within boxes might not be a bad idea. Possibly the safest approach to using MCRs would be that generate an MCR to a non-endogenous gene, like say GFP or something like that, or a recoded sequence within the genome, in which case the wild population doesn't have the target, so it can't go in there. And so that's a pretty fail-safe sort of method, and the confinement there, again, I would think would be back to the ACL1 level and perhaps in a standard in a standard fly room. So those are just examples of possible suggestions in terms of thinking about safety in the lab. And again, in terms of rules of thumb, I would say that perhaps if one's looking at a pioneering experiment in a new insect species, like, for example, what Valentino had set out to do at the first, maybe a good rule of thumb would be to increase the ACL level by 1. So most of the insects, for example, that people would be interested in doing anything with would not be vectors for disease, and so that would just boost it up from ACL1 to ACL2, as we did with our fruit flies. If adequate biological confinement strategies exist, like splitting the Cas9 away from the gRNA or using a recoded or exogenous sequence, then in principle, in discussion with a biosafety committee, the ACL level perhaps could be reduced. And I think my strongest suggestion would be that that as the guidelines go forward, that we continue with the well-vetted practice of relying on existing institutional biosafety committees which of course involves seeking guidance from the NIH or other federal agencies when necessary, to judge the safety of experimental protocols on a case-by-case basis. Having gone through a very, very extensive review here at UCSD in this way, I have a high faith in this type of process, and I believe that this is this policy, which has been a linchpin of biosafety review in the past, should continue. It's a very successful method, in my view. Now, I'd like just to briefly touch in the last few minutes here on this idea of reversal elements. We have a set of elements that we've been studying in the lab, which we refer to as erasers, elements for reversing the autocatalytic chain reaction, that in principle would be able to freeze the spread of an MCR element once it had been released, say, into a wild population, and reverse that spread or prevent the further spread of it. And so there are several different implementations that we're testing of this idea, but the basic idea, again, is that an MCR would be released into a population over, say, 10 generations or so, or released at 1%. It would go to saturation within the population, and then at that point one would release an eraser. The eraser then would be such that it would spread within the population of the MCR population, but not on its own, and then would go to completion, taking down at the same time the MCR. And so, in principle, that would be a way to prevent the further spread of the MCR. And I know there's been a lot of talk about reversal constructs, and again, we are very actively working on this. And they could be used to carry, the erasers too could be used to carry antimalarial infectants. So that at the end, for example, when one is at this point, one could have now erasers that carry the antimalarial cassettes. The MCR that initially drove the whole process into the population is now gone or close to gone, hopefully. And then, you know, you have a population that's kind of frozen, but that everybody, at least at that time, has the cassette. And then the advantage of that, of course, is that there would be no further expansion of the range. So for one issue that has certainly arisen as an issue to think about moving forward into the regulation of this in the field is that you could easily have a situation where you have two countries, one which feels comfortable with using genetically modified insects and one which does not. And so if in the one that does, if you release the organisms according to their guidelines, then the risk would be is it crossed the border, and that could cause trouble and political instability and whatever, which would be a serious issue, I think, a geopolitical issue to consider. And so, this might help prevent that. But the negative there, of course, is that if you stop the MCR, then now new mosquitoes coming in from the edges will no longer get the element, and so one might have to do it again with the MCR and the eraser. So just a few points to consider regarding the use of these reversal elements is, are they really necessary in all cases? Like, for example, in suppression schemes where the point is you want to get rid of the organism anyway, do you really need to have an eraser in those cases? And that's something to consider. The other, I think, which is the one that I would be most focused on, is what are the risks of deploying reversal constructs? So how might they interact with MCRs and wild-type alleles in various scenarios, for example? We've been actively collaborating with Fred Gould and doing modeling on these, and although the experiments, those modeling tests so far runs have been very preliminary, they have already revealed complex behaviors between wild-type MCR and eraser alleles that none of us had originally anticipated. So that's something to bear in mind. You definitely want to do that modeling very carefully. Consider what kinds of weird interactions can take place once you have all these different kinds of alleles present. You also have MCR alleles that are, you have alleles that get generated by the MCR that are done by NHEJ that are resistant to MCR by just creating a mutation in the site. So now you have to wonder about how those alleles also interact with all the others. And those are actually much more complicated than one might have immediately expected. The other is that the reversal construct may also, by virtue of acting on the MCR, may create unexpected drive elements that weren't anticipated. And in fact, in our own initial experiments on this, we have run into this as a very significant issue. Now, we believe we understand the basis for why that's happening and are able to, we think we are able to at least logically counteract those effects. But we still have to test those and see if, in fact, it is possible to get erasers that work very cleanly. But even if they do, in initial lab experiments, one has to always worry that there will be things that happen as they go into nature. And Hamid Malik, for example, has also, in a blog, has written to this effect that one might want to be concerned about the possibility of erasers not, or reversal elements not working exactly as intended. But again, I think with more empirical data accumulating, it'll be easier to judge that situation. And then there are, the other consideration too is just what other strategies are out there for counteracting a released MCR, should you want to recall it or mitigate some effect from it. And of course, an obvious thing is just to maintain a population of wild-type mosquitoes, and then that eventually they could be used to repopulate an area if, for example, a species had been locally driven to eradication and you didn't want that to happen. Another thing would be to create a population of immunized mosquitoes, that is, they had recoded targets and then release them. Of course, the negative there is that you're modifying the population by doing that, but that would be another alternative. So those are just some considerations to think about. And so some kind of final considerations that I'd like to leave you with then is, again, having gone through this consensus building process, which, you know, at a very, very small scale is a sort of reflection of the process in biology that was started back in the famous Silmar conference back in the 70s, which one can then look back now perhaps at that and look at the successes and limitations of that first sort of serious introspection on the part of the biological community, in that case through the use of recombinant DNA. And of course the successes that I think are difficult to underestimate, very valuable, is it established a tradition of transparency in the biological sciences. And I think that is absolutely critical and very, very important to maintain. And it provided a new framework also for ethical discussion between scientists and the public, exactly the kind of thing that's going on here. Again, extremely valuable. The limitations, though, is certainly as I've been reading more about it, is that it was difficult to achieve a strong consensus view, and in the end it was scrambled for very desperately at the end. And subsequently, federal policies that were adopted in light of the discussion that had gone on there were quite stringent and ultimately judged to be too prohibitive. And it has been argued that in some cases experiments like, for example, on oncogenes were delayed by as much as 4 years. That is perhaps a cost that maybe was not necessary to pay. So what I would suggest, and this is just my own opinion, to achieve a balance between mitigating risk and impeding research is that, of course, one considers strategies to reduce risk of laboratory and field experiments. And again, is one possible starting point for that. The consensus piece we all put out would be maybe laying out some of the ways that's done, so that we could establish responsible guidelines for safety of laboratory and field experiments. Field experiments as well, of course, which is an add-on that we didn't consider in that particular document. And then my own personal plea would be that one maintains a reliance on IBCs, that is, the Institutional Biosafety Review Committees, to judge the safety of experiments on a case-by-case basis. I can't emphasize that enough from my own point of view, having gone through this process. And two, to consider the risk also on the flip side of the coin of impeding gene drive research, perhaps by adopting measures that are perhaps too restrictive. And this would, I would say, also go for the implementation in the field. So I would say with regard to the laboratory work, it's absolutely essential, I believe, based on our own work, for example, with reversal drives in the lab showing that they do have some defects that really do need to be addressed, that one has free access to do important experiments, again, with the approval of an IBC. And that the other issue, too, is that if one adopts perhaps too onerous a regulations, other groups might just be scared off from doing it, and it would kind of favor those with irons already in the fire that already have pre-approved protocols and so forth. That was an objection that was actually also raised at the time of the Silmar meeting. And finally, that impediments to research and field deployment could delay it for important applications. The one I gave an example of was oncogenes, which was made as a very salient point by James Watson in the defense of DNA. So I think if that's the kind of thing I would suggest in the end, as my final sort of leading recommendation would be to try to, and I know this is a very difficult thing that you're all undoubtedly struggling with, but to achieve a balance between mitigating risk and impeding important research as it moves forward, because there are very, very important things that this technology offers. So with that, I'd like to point out, of course, that Valentino, who is here with me and can answer questions, was the man who came up with this idea and made it work. And then, in addition, we had very valuable advisors at UCSD who helped us frame this problem to begin with, consider what the, how to mitigate risk and proceed, as well as Hugo Bellin from Baylor, who was very valuable in that as well. And Joe Vinitz at UCSD, who has kindly provided the integrity in which we did this work, and the collaborators that we've established a variety of collaborations with moving things forward. So with that, I'd be happy with Valentino here to take questions. Thank you.
Thank you so much.
That's terrific. Keegan, do you want to take it from here, or do you want me to just do it from Arizona?
Go right ahead.
All right, well then let's open it up. First, thanks very much for a wonderful presentation. Really, really quite clear, and you covered lots of really interesting material. So once again, this is Jim Collins, and let me open it up to my colleagues on the committee who might have questions for you and Valentino Gantz. Committee members?
This is Greg. Can I be heard? Am I unmuted?
I can hear you, Greg.
Oh, wonderful. Okay, so thank you for a wonderful presentation, Ethan. My question had to do with your discussion of the risks of the reversing constructs. I was trying to, it sounded, I gather that with a reversing construct there would be additional risks above and beyond whatever are incurred by the MCR, the original MCR. But I was trying to figure out whether the additional risks are simply additive or are they sort of multiplicative? Are they, you seem to be suggesting that the reversing construct, the eraser, introduces a level of complexity that it's not just that you have, if you think of the eraser as itself an MCR, it's not just that you now have two MCRs out there, but that you have so many changes going on that there's very dramatic reasons for concern. Am I making sense? And is that right?
Well, I'll give you my take on it, and then maybe Valentino can give you his. I guess what I mean to say there is that if one just considers the complexity, you have the MCR, you have MCR-resistant alleles, you have the wild-type allele, you have the eraser, you have eraser-resistant alleles that can pop up in principle. And these are all interacting in ways where, in the wild at least, they might not be like they would in a lab where you have everything in a box. So you could have pockets of organisms that, say, get the MCR but don't get the eraser for a while. So you could have very complex population-type interactions. And again, just in the initial modeling analysis that we've been doing with Fred Gould, it's been extremely educational and illuminating in my case, in our case here, just to see what they can do. And then one, of course, has to imagine that for each of those different alleles, they're associated with a particular fitness. They may not all be equally fit to the wild-type allele. So all I'm saying is that there's something inherently unpredictable about that, and that one needs to certainly consider the range of things that one might expect, which again can span some interesting phenomena, and ask, well, is this okay? Is this tolerable? Are there fixes for it? And so forth. And then, of course, the other thing that I think, too, is not completely easy to eliminate is the idea that once you have all these interactions, that you might, again, without not fully understanding, at least at the beginning, how it might happen, although we do know already how it can happen, is that you might get other types of drives that you hadn't intended to make that just come from the interaction of these active genetic elements. And so I think one has to be pretty careful and really understand all of those interactions as best as possible before thinking that it's necessarily a good thing to put the eraser in after the MCR. And, the other thing I think, too, is if you say, well, look, if your strategy is a good strategy of using the MCR, maybe the best thing, at least in some cases, is just to leave well enough alone, and then at least you're dealing with a fairly simple genetic situation to monitor. And there are alternatives to using reversal elements that, again, I think deserve some consideration. Valentino, would you want to add anything to that based on your view of the whole thing?
Yeah, so on top of the interaction that this allele and construct might have in the wild, like from the first results that we've had, we've seen that there might be some interactions between how an eraser goes and replaces an MCR by creating some events, like recombination events that we were not foreseeing. So I'm working currently into trying to generate a second version of this construct that would avoid this first set of problems that we've observed. And we are already envisioning a third version to try to avoid some other problems that we think might be arising. Does that answer your question?
Yeah, I think so. It sounds to me like it's not just an additive problem. It's an interaction. It's a multiplying. There's a multiplying effect.
Right, and I think that another thing is just inherently somewhat unpredictable. I think that's the thing to worry about. I mean, at some point we may be able to predict them, but based on what we know now, we haven't yet been so successful in predicting, you know, sort of first-order possibilities.
Thank you.
Hi, this is Lisa Tannehill. Can I ask a quick question?
Sure, Lisa, go ahead.
I was just wondering if you could comment a little bit more about the ability of the carbon copy element or the split gene drive to really act as a gene drive, because I'm trying to wrap my head around that from a genetic standpoint, and I was wondering if you could just talk a little bit about using that technology as opposed to, say, an intact gene drive and sort of the advantages and disadvantages of that.
Okay, I guess my feeling is that it probably would not be a very effective gene drive to try to use on purpose. And the reason is because it's acting in an additive linear fashion. If it faces even a tiny bit of negative selection, it'll succumb to it because negative selection acts in an exponential way. So I think if you really want to make a, you know, and my feeling is, you know, again, talking to several different people in the field, is that although one does want to be very, very cautious and think this thing through, most likely when it really comes down to field implementation, none of these systems are going to work as well as we hope they will. So I think something like a copycat split drive element, it'll just fail, I think.
Okay, thanks a lot. I appreciate the your comments.
Hello, Jim. This is Nicole. Can you hear me?
Yes. Go ahead, Nicole.
Yes, I have one question. So again, Ethan, great presentation. You mentioned, you know, the fact that there may be interactions that, of course, have some level of uncertainty around that. I'm curious to know, has there been a discussion on what is the thresholds of efficacy by which one who's using this science may decide that it needs to shift gears a little bit for the next round of experiments? In other words, we need to set some standards, I would think, for what is going to be our decision point. Has there been discussion around minimum levels of efficacy and setting those thresholds?
I'm not entirely sure I understand your question. Are you suggesting, for example, that you'd have to have an MCR that went above 95% before you thought of using it in the field versus 70%? Is that the kind of question you're thinking of?
Yes, exactly.
Well, there again, you know, I probably want to defer to an expert like Fred Gould on that. And, you know, we are doing the modeling that does include varying that parameter over a wide variety of values. And again, you know, one wants to look at the modeling. Obviously, the lower the drive, the more time it will take, the more generations will be necessary for complete takeover of a given population. On the other hand, as long as the drive exceeds the negative selection against it, it should be a drive, and it should eventually be effective in an ideal situation. And I think, you know, it's hard to know. My guess is that the real answer to your question will be an empirical one, that people will try drives, at least in case studies in the field, that function with different efficiencies, and ask, you know, what is the lowest level of efficiency where you have a prayer of having it work. But I would think, just by common sense, that the more efficient that it is, the more likely it is to accomplish its goal.
Thank you very much.
Thanks. Ethan, this is Jim Collins. Just a quick question to cycle back to the containment. At the moment, is all the containment using insects? Is it voluntary, or does it carry the same kind of regulations as work does with vertebrates, for example. Can you talk a little bit about that?
You know, the truth is I don't know. You know, of course, there's a general requirement for all genetically modified organisms to be maintained under confinement in the laboratory, but that's, you know, pretty loosely followed and monitored. Beyond that, I actually don't know. I mean, we certainly took this on in a voluntary way, and we were quite proactive in talking to the local IDC about it. And, you know, I would expect that the recommendations that your committee eventually offers would be ones that would be adopted by the NIH, and at that point they would become binding. And that would be my guess as to what the trajectory is, but I think we are in that intervening time when it's unclear, and that was one of the reasons that we were all stimulated and motivated as quickly as possible to get that consensus piece out and that policy forum piece, because we felt that in that vacuum of clear regulatory guidance that it made sense to at least alert people to the risk and to ask people, of course, to use the common sense that I believe nearly all scientists have to make sure that they apply appropriate safeguards. And the good news there, too, is I think that there are plenty of very effective methods that, when used in you know, various combinations, are undoubtedly going to prove to be very effective. But the answer to your question is I don't know.
Yeah, I mean, I know of institutional review boards, cases of institutional review boards, which have said, well, look, we just don't do invertebrates, so you don't even need to bring the issue to us. And or on the other side, investigators who would say, review boards don't do vertebrates, so we don't even have to bother. Right,
and again, I would hope that that wouldn't be the standard view of an IBC now that there's been enough discussion about this. If you know of examples of where that's actually happened, I'd actually be kind of interested to know about that, and I would be happy to discuss that with our own IBC in terms of how they interact among themselves. Certainly within California, there's been quite a bit of conversation between the various campuses.
Yeah. Great. Thank you. Thanks.
Welcome.
Other questions from committee members?
Jim, I just had one more. Oh, sorry.
No, go ahead.
Hi, this is Lisa again. I just had one more question for you all. In light of the different technologies that are out there to do sort of genetic engineering and for establishing these gene drives, do you think that the CRISPR-Cas9 system is going to be the one that the majority of people will employ?
I think so. And again, you know, it's hard to envision the future with a field that's moving this quickly. There are some defects with the zinc finger proteins. They have repeats in them, so they're not as stable. And you know, it's very difficult to beat the programmability of CRISPR, where, you know, all you have to do is build a little gRNA. And now the new CPF1 that was just published by published in Cell last month offers a potential to increase potentially the frequency of homology-driven repair, which of course is necessary for this drive process because it cuts at a distance from the binding site. And the reason why that might be important is that at the moment, one of the limitations that is a potential problem, and again, in the modeling, it comes out that can indeed be the case, is that you know, you get these non-homologous end-joining mutations that then prevent subsequent conversion because you eliminate the site by which Cas9 and the gRNA would cut. That's much less likely with CPF1 because you can cut and cut and cut with NHEJ, and as long as you don't make a really big lesion, then you can go on cutting until presumably you might get an HDR event intervening. So, you know, we haven't had a chance yet to look at that, and I'm sure others will as well, but that would be a possibility. And I think, you know, given the programmability and the new forms of Cas9 and Cas9-related proteins that are coming in, my guess is that that will be. The negative, I suppose, of the CRISPR system that everybody, of course, also is aware of are the off-target effects, and it could be that there are other systems that will emerge that have good programmability that have less of an issue there. But there again, my expectation is that the CRISPR field, moving as quickly as it will solve through NIC-ACEs and other innovations that are yet to come, will solve that problem or mitigate it to a point where it's no longer a significant concern, or it's a concern that can be addressed, that will be addressable, let's say.
Great, thank you so much.